Instructions for making proteins are coded by segments of DNA called genes. But proteins are not made in the nucleus. Instead, a strand of ribonucleic acid, or RNA, must carry the instructions for making proteins out of the nucleus. This type of RNA is called messenger RNA. Both DNA and RNA are long chains of nucleotides. Each nucleotide contains a sugar, a phosphate group, and one of 4 bases. RNA is different from DNA in 3 important ways. First, RNA is a single strand. Second, RNA has a different type of sugar called ribose in its nucleotides. Third, RNA contains the base uracil instead of thymine. The other bases found in RNA are the same as in DNA. They are cytosine, guanine, and adenine. The manufacture of RNA from a DNA template is called transcription. Transcription begins when the 2 strands of DNA in a specific region unwind and separate. Only one strand of the DNA serves as a template for the new RNA. The enzyme RNA polymerase matches RNA nucleotides with the complementary ones on the template strand of DNA. Recall that RNA contains uracil instead of thymine. Therefore, uracil pairs with adenine on the DNA strand. Transcription continues. C is paired with G. A is paired with T on the DNA strand. And U is paired with A in RNA. As the nucleotides are assembled in the proper sequence, RNA polymerase moves along the DNA strand, assembling the messenger RNA, until the entire gene has been transcribed. Hydrogen bonds reform between the complementary bases in the DNA molecule, reattaching the 2 strands. The coded instructions on the DNA molecule have been copied to the newly manufactured strand of messenger RNA.
Transcription of DNA. The process of transcription begins as enzymes unzip the molecule of DNA, just as they do during DNA replication. As the DNA molecule unzips, free RNA nucleotides pair with complementary DNA nucleotides on one of the DNA strands. Thus, if a sequence of codons on the DNA strand were AGC, TAA, CCG, the sequence of codons on the RNA strand would be UCG, AUU, GGC. When the process of base pairing is completed, the mRNA molecule breaks away as the DNA strands rejoin. The mRNA leaves the nucleus and enters the cytoplasm.
A protein is a chain of amino acids. The process of linking amino acids together to make a protein is called protein synthesis. Protein synthesis occurs in a cell's cytoplasm. mRNA leaves the nucleus through a nuclear pore after it is transcribed from DNA. The assembly of protein in the cytoplasm takes place on ribosomes. Ribosomes consist of 2 subunits, a small ribosomal subunit and a large ribosomal subunit. Floating within the cytoplasm are amino acids and transfer RNAs, or tRNAs. Each kind of amino acid binds to one specific type of tRNA. Notice the 3-base region on the tRNA. It is complementary to a 3-base region on the mRNA. Each group of 3 bases on the mRNA is called a codon. The complementary 3-base sequence on the tRNA is called the anticodon. A specific tRNA carrying the amino acid methionine binds to the mRNA. The anticodon on the tRNA matches the codon on the mRNA. A second tRNA with its attached amino acid binds to the second codon of the mRNA. The first amino acid is transferred from its tRNA to the second amino acid, and a bond between the two amino acids is formed. The protein chain has begun to grow. The first tRNA detaches from the mRNA. The ribosome moves down the mRNA to the third codon. This codon is recognized by the anticodon of another tRNA carrying an amino acid. The growing protein chain is transferred to the third amino acid. The second tRNA detaches. The ribosome again moves along the mRNA. A codon is exposed. Again, the codon is matched by the anticodon of a tRNA carrying an amino acid. The protein chain is shifted, adding another amino acid to the chain. The tRNA that gave up its amino acid detaches, and the ribosome moves down the mRNA strand to the next codon. This process, called elongation, builds proteins one amino acid at a time. The 3 bases that make up a codon determine which tRNA attaches. Since each tRNA carries a specific amino acid, the sequence of amino acids in the new protein is determined by the sequence of bases in the mRNA. The ribosome continues to move along the mRNA. At the end of the mRNA, the ribosome encounters a stop codon, such as UGA. The completed protein is released. The tRNA floats off, and the mRNA detaches from the ribosome. The ribosome splits into small and large subunits.
If we could journey inside ourselves, into a cell, we would see 23 pairs of chromosomes packed into a nucleus. Each chromosome contains a long coil of DNA. If all the chromosomes were unwound, the DNA in just one of our cells would stretch 6 feet long. The DNA double helix contains 4 kinds of building blocks. An A always pairs with a T, a C with a G. DNA contains information to make every part of our bodies with its 4-letter language. Each of our thousands of genes codes for a specific part. RNA polymerase copies the information in a gene into a messenger molecule, messenger RNA. The building blocks of messenger RNA and DNA are called bases. The bases on one strand of the DNA specify the order of bases on the new strand of messenger RNA. The DNA always stays inside the nucleus, but messenger RNA travels out into the cytoplasm. There, a protein-making machine called a ribosome can read messenger RNA to make a particular protein. Every 3 bases of the messenger RNA molecule codes for an amino acid. Proteins are made of amino acids. tRNA molecules help translate the language of DNA and RNA into the language of proteins. tRNA molecules bring the right amino acids that the ribosome links together to make a protein. Proteins are the laborers. Some form structures like tendons and hair. Others perceive light, scents, and flavors, control chemical reactions, and carry messages between cells. To understand our molecular selves, scientists have read the 3 billion letters making up the DNA in the human genome. Different sets of genes interacting with complex environmental factors influence things like our looks, personalities, and risks for diseases like cancer and heart disease.